I'm going to introduce our two speakers. The first, Dr. Nir Barzilai. Dr. Barzilai is the director of the Institute for Aging Research at the Albert Einstein College of Medicine and the Ingeborg and Ira Leon Rennert Professor of Aging Research. He's also professor of medicine, professor of molecular genetics, and a member of the Diabetes Research and Training Center, where he directs the physiology core, and he's also co-director of the Montefiore Hospital Diabetes Clinic. Dr. Barzilai's interests focus on basic mechanisms in the biology of aging, including both the nutritional and genetic determinants of lifespan. He has discovered the first longevity gene in humans and is further characterizing the phenotype and genotype of humans with exceptional longevity through an NIH-supported program project. Dr. Barzilai has published over 140 peer-reviewed papers, reviews, and chapters in textbooks. He's an advisor to the National Institutes of Health on several projects and initiatives, and is a member of the Aging Systems and Geriatrics Study Section. Serves on several editorial boards and is the reviewer for numerous other journals. And Dr. Barzilai has received such prestigious awards as the Beeson Fellowship for Aging Research, the Senior Ellison Foundation Award, and the NIA Nathan Schock Award for his contributions in elucidating metabolic and genetic mechanisms of aging. In his capacity as the director of the Institute for Aging Research at Einstein, he leads 3 large program grants on the biology of aging, additional individual grants, and a training grant. And you can see he keeps busy, and I think maybe one of the reasons that Nir can think in this bidirectional way is that he grew up reading not only left to right, but right to left. That may be an advantage.
You got it. Thank you, Paul. That was going to be my first sentence. Thank you so much. So I know that we're sitting in this new and wonderful hall, but we all know that life for most of us can be depicted in this double-deck toilet. And at Einstein, we're very strong with the theories of relativity, and it's not hard to imagine that you better sit here and not here. And I'm saying that for 2 reasons. One of that is to really congratulate the leadership of the CTSA, Harry and Paul and Briar and everybody else, because all my interactions through my life here at Einstein, what happened was that they're on the bottom and I'm on the top. And they were absolutely there to receive all my complaints and griefs, and I really thank you for being receptacles. The second thing is, just because I'm going to talk about aging, I want to just give a brief introduction that's really depicted in the story of the old couple, and the wife turns to the man and says, "Honey, why don't we go upstairs and make love?" And the husband said, "Sweetie, I cannot do both." Okay? So that's the definition of aging. And I want to tell you briefly of our attempt to really look at the genetics of human longevity. And when we started it, it was a very simple thought. And the thought was that only 1 out of 10,000 gets to the age of 100. So this is a very rare phenotype. So it's more rare than cystic fibrosis or monogenetic disease. So why don't we just try and look at the genetic of those people? And so far, we recruited 450 people in the range of 95 to 110, average age 100, and we started characterizing them in many ways. In the meantime, others and us have showed that those people have remarkable history of exceptional longevity in all parts of their family. And the 2 things together made us think that maybe there are inherited protective factors that can assure humans longevity, and that's what we try to focus on. So let me tell you briefly about those 450 people. 4 of them are depicted here. They're all siblings. And actually, this woman here is now 106, and everybody else in her family is above age 100 or soon will be. And they're all healthy, and we ask them, as we ask everybody else, many questions, but also, why do you think you live to be so old? And here we usually get 2 answers. One answer is, hey, you know, all my siblings are alive. It's in the family. Or my mother was 102, my grandfather was 108. So there's this story. And then we come and say, okay, now tell us the truth. You know, tell us your secret. You ate yogurt all your life. You're a vegetarian. And it's really amazing that 450 cases later, I don't have anything to tell you. I don't have a yogurt eater, no vegetarians. 30% of them were obese or overweight in the 1950s in the United States, where nobody was really overweight in the 1950s. Nobody has exercised. Nobody was an athlete. This is a picture. This is around of the second longevity gene that we uncovered, and so you understand that this is posing to a picture that was taken in the early 1900s. And when this brother got his gun, she got permission to start smoking. It was very ladylike in New York of the 1900s, and she celebrated almost 92 years of 2 packs of cigarette smoking. And 30% of our subjects have been smoking for over 40 years. The point I want to make without showing the data here is, on one hand, they had family history of exceptional longevity. On the other hand, as a population, they didn't interact with the environment the way we tell our people to interact with the environment. Okay, but I'm telling you that I have a bunch of centenarians, and obviously the question is, what is the scientific experiment here? What is the appropriate control group? After all, the people who were born with, at that time, by the way, life expectancy for those people was 40 at birth. Impacted largely because of epidemic of childhood diseases. If they got to 40, they live to 60. But the point is that their cohort has died 40, 50 years ago. So what can we take as a control? And what does it really mean if we take a blood sample from 100 years old? Let's leave aside the DNA for a second. What is the phenotype going to tell us? We can measure something that's high, and maybe it was high all their life and was relevant to their longevity, but maybe it actually was low all their life and now at the end of life, and they're at the end of life when they're 100. So what is the phenotype going to tell us? And in order to overcome that, what we decided to do is to actually recruit only centenarians where we can recruit also their offspring. And for the offspring, we hypothesized that they're going to be enriched with the longevity phenotype and genotype, and the advantage is for them we can match age and gender match control. But by the way, for the clinical investigators, we went to the CCI. I had a cover letter that I'm going to look at centenarians and their children, and at the next day the package came to my desk saying, for children you need other forms, okay? No, they're 70 years old and they can take care of themselves. And the last point is, when you want to do genetic discoveries, you're trying to do it in as homogeneous population as you can. I think Icelandics are the best population with the most outcomes from genetic perspective, because there are 500,000 people that are all children of 5 Vikings and 4 Irish women or something like that. So, and Ashkenazi Jews, because of a long history, is really a good population for genetic discovery. Now, to just show you that we're probably right in our approach, we looked at age-related diseases, comparing age-match offspring of centenarians to control in red, and looking at prevalence of several diseases, and you see that they have less hypertension, less diabetes, less myocardial infarction and stroke. So they are healthier at this point than their offspring. But I really want to bring in the centenarians themselves, because the centenarians themselves have almost the same prevalence of hypertension, they have much less diabetes, and almost the same prevalence of myocardial infarction and stroke. So from an age-related disease perspective, their aging has been delayed by about 30 years. So this is our population. What can we do with phenotype? I'll give you a quick example, a quick but important example. You know, we have the lipoproteins, the LDL and HDL, and each one of them and each one of us comes in different sizes. And apparently the large sizes are better for a variety of reasons. So let me show you what happens to our control. It's actually our control and the Framingham offspring study where we collaborated with, and we did it in the same lab, to HDL particle size and LDL particle size. They don't change with age. Although people who are epidemiologists in the aging field will say that means that you don't get to age of 85 if you don't have large LDL particle size. But let me put in the centenarians. The centenarians have the highest lipoprotein particle size that has ever been published for a group of subjects, not individuals, but for a group of subjects. Now, you can come to me and say, what does it mean that at the end of their life, when they're maybe losing weight, they have large lipoprotein particle size? And my answer will be, look, their offspring as a group have also larger lipoprotein particle size. In fact, half of the offspring have large lipoprotein particle size. They're biphasal distribution, and so we can look at the heritability and show that the heritability of lipoprotein particle size is very significant, depending on gender and whether it's for HDL or LDL, but it's a strong heritability. So this is an example of how we deal with the phenotype, and I'll give another example. I'm not done. I just want to thank the people. I'm going to show some of the results up front. The project started as a longevity gene project, and I'm going to show data from Gilad Smon and Richard Lipton, but now it's a PO1 that has many projects, and I'm going to talk a little bit about Aviv Bergman, Yushin Su, Hassi Cohen from UCLA, Jean Crandall, and Ken Yee here. And there's many other collaborations, and that's really the fun of what we're doing, and we have to do more of that. Some of you mentioned Francis Collins. We're doing a lot of collaboration these days. So the point is that there are lots of people to do this kind of research. So from a genetic point of view, how do we dare to go into the most complex condition? I mean, isn't it hard enough to find genes for obesity, diabetes, cardiovascular disease? And in the field, there are two approaches. One of them are people who think relatively simple, and I'll give an example, and some who think a little bit more complicated, like I do, but have actually a solution to them. So let me start with the simple approach to aging. You know, ponies live longer than horses, and small dogs live longer than big dogs. If you knock out the IGF receptor, insulin IGF receptor in nematodes, they become abdominally obese, but they actually live longer. If you have some models that spontaneously are dwarf, or that you made them dwarf by knocking out the growth hormone or causing low growth hormone IGF level or action, they all live longer. So in nature, across models, being small is associated with longevity. And the way for us to approach, if this is important in humans and to our centenarians, was to start with a phenotype, so start with the bedside, and look at, first of all, IGF level in our control and offspring male, and then in our control and offspring female. And interestingly, the offspring female had higher IGF levels. But we had another clinical thing, and this is their height, the maximal height of those people. And the males had the same height, whether they were control and offspring, but the females were significantly shorter. So what could explain the fact that our population have higher IGF level, but they were shorter? It's remindful of the model that is IGF receptor heterozygous knockout, and they do live longer, they are smaller, they're slightly smaller, and they have higher level of IGF. And it happens in females, and in males there's just a mild effect, but it's not significant. So based on this phenotype and based on what happened, what people came out from the bench, we started with Yushin Su, who was then in San Antonio. Now she's recruited here. Yushin Su has a great technology to practically sequence every gene of interest. It's practically, but it's really very good. And what she's done is she found some old mutation on the IGF receptor and some new mutation in the IGF receptor that she found in our Jewish population. But those mutations were only in 7 centenarians. Actually, 7 of the centenarians had 2 of those mutations. And the question is, are those mutations of functional significance? And this is in collaboration with Hassi Cohen, and actually we've done, and Yushin has done it in other ways, but what we took is we took the transfer lymphocytes from our patients, and that's the tissue that we can do things with, expressions and function. And Hassi described that those lymphoblasts have decreased IGF receptor, and when he incubated those T lymphocytes with IGF, there was a significant decrease in phospho-AKT phosphorylation. This and other things supported that those are functional receptor. Okay, so this is a simple way to come from the people who have simple models, one gene, longevity, and show if it can test it in centenarians or not. But it accounted for only less than 2% of our population, and we're trying to find significant genes that are more of public interest. And this is how we go about it. And this is why, although it's complicated in aging, we have an opportunity that nobody else has in the complex field. And the opportunity comes from the fact that between ages 60 to 100, you know what happens? Almost everybody dies, okay? So there's a great selection here. The fact that we have 450 people between the ages 95 and 112 is the oldest, and we can populate everyone here, it's great. But we are having a great selection. In the United States, half of the people die at the age of 78. So if we have a genotype, and when we put the frequency of the genotype on the age axis, and it looks like this, which means it's underrepresented with age, it's monotonically decreased, underrepresented at age, we think it's an aging or killing genes because it's not presented anymore in the people who are surviving. And we're interested in that. But we're much more interested in another genotype, and this will be a genotype that's relatively rare at age 60, but it's monotonically increased and it's overrepresented in centenarians, and we would call that a longevity gene. These lines of monotonic increase and decrease provided us with a whole different statistical approach, because we think we're going to deal with false positive much better. Because if one on the line will be higher and then it will go lower, it must be a mistake. It's not that you have longevity genes when you're 80 and when you're 100 you're losing it. So lots of other things that I'm not going to go into are very helpful in looking at this approach. And of course, most of the genes are not going to contribute to lifespan. So far, we discovered and validated, or we didn't validate, but there's at least one validation outside of our lab to those 3 longevity genes. And I'll tell you just very shortly about them. 2 of them are lipoproteins, and they are actually related to the lipoprotein particle size. In particular, CETP is known to regulate lipoprotein particle size and HDL level. And both of the homozygosities, so the homozygosity here is in an exon. The homozygosity here is in the promoter. And here it's a deletion in the 3' untranslated region of the gene that has a lot to do with the secretion of the adiponectin gene. And I think the most important thing to note is that the homozygosity is between 8 and 12% at 60, and it's between 24 and 32% at age of 105. So this increase and this overrepresentation is really quite huge in a genetic term, or if we compare it to, for instance, the new genes that were found to diabetes. Those are much more impressive and they're much more public. Also to note that in each case, we can measure the genotype in the plasma. It's not a knockout or a hyper or a huge hyper doubling of something, but it's a 30 to 40% decrease in CTP ApoC and an increase in the adiponectin phenotype according to the genotype. So they're all significant. We moved from those were part of candidate gene approach, and we have completed our first batch of 600 subject using the Affymetrix 6 technology that has 1.6 SNPs and CVNs on the chip. So it's a huge amount of information, and we're very happy and capable in dealing with all this information. And so far, we discovered 15 longevity genes and 46 aging genes with a p-value of at least 10 to the minus 6. You know, the more genotypes you have, the more false positive you have, the more stringent the genotype criteria is going to be. So this is our criteria. And just to show you, those 15 are kind of interesting because they are kind of have low presentation in the population, but they almost, each one of us is almost double. At least those are also almost doubled in centenarians, the frequency of those genotypes. And the second thing that's really important is that you can look at all of those. If it means something to any one of you, let me know. But those are, as far as we know, totally new candidate. Nobody has talked about anything of those in relationship to aging. And I think this is the exciting thing. And this is what we hope to now go, you know, to the bedside and back to model and understand what are those genotypes and are they really associated with aging itself. Some of those trends that I told you about, the first two that we're looking at, and also some of the aging diseases and their trends, and it's all at the 10 to the minus 6, 10 to the minus 13 level. One of the things, so you notice for every 15 longevity genes, we have maybe 45 aging genes, which makes you think that each longevity gene protects against more than one disease. And we actually published the CTP. When we published the CTP genotype, we showed that it's protective against the metabolic syndrome and cardiovascular disease. But what we try to do is to see if the centenarians are centenarians. Some of them have really great cognitive function. They have great mini-mental state exam. It's a 30-point, and they get over 25, and the others are all over the scale. And we try to see what does it take for centenarians to get with his mind and not only with his body. And one of the things that we discovered only for the CTP genotype is that those with better cognitive function have 3 times more of this genotype. This genotype came up not only in the cardiovascular, but also in the cognitive field. And not only that, with the collaboration of Richard Lipton and the Einstein Aging Study, they have a prospective study of people who are becoming demented or not. He showed that people who develop dementia have 20% of the frequency of the genotype of the non-demented people. And we're working hard about this. It has a lot of implication, including therapies. I want to close doing several things. This is Frida, my grandmother-in-law. She's the grandmother of my wife, Laura, and she died 2 months ago at the age 102. And she was an inspiration for me, and the joke that you heard before about the older couple, all those geriatric jokes come from her, and I have a lot of them, so I don't have to repeat it. And this is her 100th birthday, and she's basically drinking resveratrol from the bottle, but I'm not sure how it contributed to her longevity. Until recently, she was dating, but only people who had cars. And they became young and a little bit annoying to her. And when she was 100, her son says, "Mom, you know, I can write to the president. He can send you a letter about your great achievement." And she said, "This president? I don't want a letter from you." So her son-in-law, my father-in-law, sent a letter to Bill Clinton and said, "That's what my mother said." And Bill Clinton wrote a letter, attached a picture which she had above her bed, and that was the love of Grandma Frida. It's JFK. It's probably not JFK who said it. You know, you discover that the presidents who are saying things, there's other president or somebody else that was saying it before. But anyhow, JFK did say it. I don't know if he's the first to say that there's not enough for a great nation to have added new years to life, but we must add new life to those years. And really the objective, our objectives, is to develop a drug that will prevent the chronic debilitating age-related diseases. The longevity for us is more of a side effect that the drug will do. Why am I very optimistic that this is going to happen? Because when Barack Obama gave his acceptance speech, did he talk about male reproduction? Did he talk about diabetes? Did he talk about cancer? No. He talked about a 106-year-old woman.
Cooper is 106 years old. She was born just a generation past slavery, a time when there were no cars on the road or planes in the sky, when someone like her couldn't vote for two reasons: because she was a woman and because of the color of her skin. And tonight, I think about all that she's seen throughout her century in America. The heartache and the hope, the struggle and the progress. The times we were told that we can't, and the people who pressed on with that American creed, yes, we can.
I feel nothing but relief that things have changed as much as they have.
This is the incredible story that is similar to some of our centenarians. Thanks for listening.